So SNiPgenie is a program for microbial variant calling using short read data from next generation sequencing platforms. And the idea is that for certain species, we can take these short reads that are a result of genomic sequencing and align them to a reference genome, and then call variants for multiple samples. And then we can use this information to create a snip alignment depending on our filtering, and then finally produce a phylogeny showing the relatedness between each sample. So this is done for a subset of species that are relatively have a very closely related sets of strains that makes this analysis useful, such as Mycobacterium tuberculosis and a few other species. And this program is usually written as a command line tool, but this is a GUI to make the process a little bit easier and to add a few extra bits of analysis that you can do. So the layout of the GUI is relatively straightforward. On the right there's a log window for messages to appear. This part here is for a table where our samples will appear when we load them, and then on the left here there's some options which we'll have a look at. So the first thing we do when we open the program, we have a blank project, is to set the output folder where the results will appear as we do the analysis. So we do this from the settings menu. We go to set output folder, and I'm going to set this folder that I've previously created. Usually it should be empty, so the output folder appears here at the bottom. And then we want to add our fastq files that we want to analyze. So I go to file menu, and then we have some sample files here. These are all paired, so there's 2 for each sample. When I open them, we get a row for each sample with both pairs, the name and the file name, the full path of the file name here. And then it shows us the read length for each sample. So these are all 150 base pairs in length. If I right click on one of the rows, I can choose to look at the quality summary, which is a simplistic version of the type of output you'd see in say the fastqc program. It shows us the quality values across the read on average, and then the GC content. Now the first step in the workflow is alignment of the reads to the reference genome. So to do that we have to set the reference genome, and you can arbitrarily pick any reference genome that suits you by downloading the FASTA file and then loading the reference sequence from here. And you can also, if you wish, although it's optional, choose an annotation file in GenBank format, which is not essential. It's used for consequence calling later on if that's needed. But there's also a set of preset genomes that are inbuilt into the program, because that's what this was developed for. This originally was developed to do analysis usually on Mycobacterium bovis. So we can set this one here because these reads are from bovis. So what will happen is then the reference sequence information appears on the left here. There's also automatically the annotation loaded and a mask file that we use, which is a bed format. So they're all built in, and there's other presets for TB, MTB, and MAP, and MS-Magmatis. Then we can just simply align the reads by selecting the reads the samples we wish to align. So before we do that, we can set the number of threads we want to use, depending on your computer. Say I select say 14 threads here. That will speed things up a bit, and that's really it. There are a series of aligners that can be used depending on what's installed in your system. This program is being run on Ubuntu Linux, so on the Linux system you should have installed those yourself with your package manager, which is straightforward. So on Ubuntu you just use apt. By default BWA will be used as the aligner, short read aligner. If you're processing long reads such as Oxford Nanopore, you should set the aligner as Minimap2 and the platform to ONT. These are Illumina reads, so I'm going to use BWA, and I'm going to just align one row, the top row here. So I can just press this button and the alignment will start. And the reason why this is very quick is that these are in fact, this has already been done in the previous run. Like I said, I used this output folder before, so they're already there, and by default the program won't overwrite previously aligned samples. And when this is done, if I want to overwrite, redo the alignments, I can just choose overwrite here, or you can go to the folder and manually delete the file. So we'll have a look at the output folder here in a minute, but first of all, to note that the BAM file, resulting BAM file from the alignment, is also added to the table, and now I can read, have a look at the read alignments using this rudimentary alignment viewer or BAM file viewer that utilizes SAM tools. So it's just a text format. So obviously if you want something more sophisticated like IGV, you can use that to open your BAM files, and we can have a look at the reads along the genome. We can also choose, if we have the annotation loaded, a specific gene. Like I said, this is very basic, but it may be useful in some situations where you want to have a quick look at your results from your alignment to see if something's wrong. And we can also have a look at the mapping statistics, which will appear here, which tells us how many total reads are mapped. So just to check the output folder, all the aligned reads are put into the mapped folder, so you can see that that's the resulting file from the first sample that we aligned. As I said, it had already been aligned from a previous run. And then we want to, we're going to align the rest of the reads in order to do variant calling, because obviously it's meaningless to do it with one sample. So I'm going to run that now. So I've aligned all those samples, so we've 4 samples aligned, and now we're going to do variant calling. And I've also saved the project so we can reopen it later if we want. And this is the slowest part of the workflow often if you've many samples. So more threads are better, as it will run the pile-up process in parallel. So the pile-up is essentially just taking all the reads at every position and looking at the nucleotides along that in that position, and then using that to call a variant against the reference genome. So you can also, before you do it, apply quality filters or change the default quality filters here. You can take this to apply a proximity filter, which excludes SNPs that are too close together. And you can also, if you provided the mask file, that will exclude the SNPs inside the regions that are in the bed file, which are genomic locations. And in MTB, say, these are regions that contain repeat regions that might cause problems with the short read aligner, and that will produce erroneous SNPs, which is why we mask them out. So I'm going to run this now using the button here, and we can see that it says calling with 4 samples, and it has happened really quickly. And the reason for that is that I'd already run the pilot before, which, as I said, is the slowest part of the process. So it's saying here that raw.bcf exists. So in order to run this and overwrite the results, we can choose the overwrite button here if we wish. And then the rest of the steps are simply post filtering steps. So and splitting the calls into SNPs and indels, and then doing consequence calling if that's needed, and then, like I say, masking, applying the mask filter. So the output folder will look like this, and the last or second last step in the process is to create the snip alignment here, and that loads this distance matrix here, which is generated from essentially from this core SNP alignment. So if you have a quick look at this file, it's simply a FASTA file for each sample with the nucleotides at each position, matching positions. So it's like an alignment where SNPs were called. So this is what distinguishes each sample for only those positions where SNPs were called. So that's why these were selected out of the entire genome. So these 4 are relatively closely related because they've only got a small number of positions that vary between all of them. And that's what's used to create the SNP distance matrix, which is this. So, and the last step is to create a phylogeny, which is a tree built using the SNP distance matrix. And this can be done with a variety of programs independently. You don't need to have this program to do it. At the moment, this program uses RaxML in the background, so you have to have that installed on your system. Again, it can be installed with your package manager if you're using Linux. And we can simply choose make phylogeny here. Again, this is quite quick because there's so few samples. And that pops up a tree in a viewer program here. And you can see immediately that these strains are very closely related to each other. The tree includes the reference genome here. And you can see the reason why they are so close together is that, say, take these 2 samples here, 19 and 15. And if we look at the distance between them, there's only 1 SNP between these samples. This is a symmetrical matrix. So that's reflected here on the tree.